Okay, let's begin today with chapter 4. Again, I've listed some of the, all of the objectives at the beginning, just to try to reinforce to you that if you're looking for the answers to these objectives, that you will find them within this PowerPoint or within this chapter, and to also kind of point out to you that if you're wondering where your test questions will come from for this unit, this would be where I would take questions from the chapter 4 portion. Okay, now most of what I'm going to talk about today is things that you should remember from Biology 103. Now, having said that, I'm also aware that many of you have not had Biology 103 for a couple of semesters, or for some of you it may have been years ago, or possibly even that you took the CLEP test and you skipped 103. Well, please remember that because 220 builds on what was in Biology 103, we do expect you to remember these things. So again, for some of you, this is going to be just review. I'm going through it fast. For others of you, you may need to spend a little more time reviewing these major cellular structures to make sure you understand what their function is. Okay, so here we go. All living things are made of cells. Okay, well, that's very, very obvious to us, right? The cell is the smallest unit of life, and so we know that things can be unicellular, made up of one cell, or multicellular, made up of more than one cell. We know that cells have basic shape. For example, they can be spherical, round, they could be cubical, they could be cylindrical. We know that every cell is going to have a membrane. It's got to have a membrane. And inside of that membrane is going to be cytoplasm or cytosol, which would be the watery portion and the organelles. And then we know that every cell has got to have some DNA. Okay, it's got to. That's the recipe on how to make another cell just like it. It's got to have some ribosomes, and a lot of students kind of forget that because this is the protein factory. And remember, DNA codes to make proteins. And they've got to have some metabolic capacity. And what that means is they've got to have some way of acquiring nutrients. Now, when we think of cells, we divide them into two major types. It's either a eukaryotic cell. Okay, and we're going to learn that that means true nucleus, or it's going to be a prokaryotic cell, which means primitive nucleus, or early nucleus, first nucleus, or before nucleus. Okay, so again, eukaryotic cells are going to have a membrane-bound organelles, and they're going to have a membrane-bound nucleus. We also know that your eukaryotic kingdoms are going to be your animal kingdom. That's the kingdom we fall into. Plantae kingdom, fungi, and your protista kingdom. Now, your prokaryotic cells are going to be your primitive or more primitive cells. These are going to have no true nucleus. And that doesn't mean they don't have DNA. Of course, they have to have DNA, but it's just not surrounded by a membrane holding it in one location. We know that the two kingdoms that fall into this are going to be your bacteria and your archaea. And remember that bacteria and archaea are also domains. And so for our purposes of classification, that's just kind of a refresher for you to remember. Okay, so remember from Biology 103 that all cells must be able to, number one, they've got to be organized. They've got to have some kind of organizational structure. They've got to be able to grow and develop. They've got to respond to stimuli. They've got to be able to transport materials in and out of the cell effectively and efficiently. They have to be able to metabolize. Okay, which means they're going to have enzymes, and we're going to refresh our memory later that you remember enzymes are usually proteins, and most of them serve as catalysts. They speed up chemical reactions. We know that cells must be able to reproduce, and remember reproduction can be asexual or sexual. And finally, we know that cells have to have some form of DNA or some kind of genetic heredity material inside of them, or else they wouldn't be able to pass that on to the next cell. So again, kind of refresher of Biology 103, that these are the characteristics of life, characteristics of cells. So again, I've already hit on this. I'll do it again. They've got to be able to reproduce. Okay, both sexually or asexually. They've got to be able to grow or develop. We expect living things to grow. They have to be able to metabolize. They have to be able to get the physical and chemical things that they need to function. They have to be able to move. It says right here irritability, and what that basically means is it's got to be able to respond to a stimulus, and that can be either positive or negative. They have to have some kind of support. Remember, they've got to have at least a membrane, but many of them have cell walls and vacuoles and other structures inside of them that kind of give them shape. And then they've got to be able to transport nutrients in and waste products out. Okay? This is a prokaryotic cell. This is a bacteria, and so it's kind of showing you some of the structures that should, make sense to you, that you should make sure you know. These would be things like, for example, the cell membrane. We see that. We clearly see the cell wall in this case. We also see that this one has a capsule, which is, an outer coating on top of that cell wall or that cell membrane. We see that they have ribosomes. Remember, they've got to have those protein factories. They have to have these things called fimbriae. And fimbriae is one of those structures. A lot of people think it's like cilia. It's not really. But fimbriae help it to stick or adhere to other cells. Some of them may actually have a slime layer. And we're going to talk about the purpose of the slime layer and what it does for the cell. Some of them, if you'll notice down here at the bottom, some of them have actin filaments. And so if you remember from your anatomy classes, actin is thin, but these are proteins that help to give them shape and structure. And if you'll remember from your muscle chapter in anatomy, you'll remember the actin myosin bridge for muscle contraction. Even though these don't have muscles, it allows for movement. Obviously, they have the DNA, the chromosomes. Some bacteria will have what's called a pili or a pilus. I've heard it pronounced both ways. And we're going to talk about its function a little later. And then again, you see the matrix. That's definitely there. Some have a flagella. We'll talk about that. And then some may actually have these things called inclusion bodies, which are like little storage tanks for things that they may need. But again, you should kind of remember these basic structures. And if you don't, you need to kind of start making sure you could identify those now. Okay, so external structures. So when we think of cells and we think of external structures, we think of appendages. Just like you and I have arms and legs and a head and things that come off of our core body, these cells do too. And the major reason for appendages is, number one, motility, which is another word for mobility, okay, motility, and that's obviously to help them move. Or it's got something that's going to help them stick or adhere, okay, to another cell, and so typically we talk about it being attachment. We also know that some external structures that a cell might have is they may have a glycocalyx, and that may be the first time you've heard that word, but that is a surface coating, and we're going to talk about the importance of that on especially your bacterial cells. Okay, flagella. All right, one of the main things about bacteria is they've got to be able to move, right? That whole thing of motility. So when we think of a prokaryotic cell, or a eukaryotic cell, both, either the one that has the true nucleus or not, you think of a flagella. Typically, a flagella has three parts. It's got something known as the filament, the hook, and the basal body. And I don't go into a lot of detail about this. I don't test you on this, but I just need you to understand that the flagella on a bacterial cell is slightly different than a flagella on a eukaryotic cell. Okay, again, both are for movement, but they are a little different in their structure. So their function is kind of the same, but their structure is a little different. We know that a flagella is going to rotate 360 degrees on a bacterial cell. So it's going to act more like a propeller. So if you think about a propeller on the boat motor, it's going to help propel the boat through the water. The flagella is going to do the same thing for your bacterial cells. I'm sorry. And how that flagella moves, or how it allows the cell to move, is often studied in microbiology to help with identification. We know that in your bacterial cells, your prokaryotic cells, it's not just that they have one flagella. Many of these organisms have more than one flagella, and where these flagellas are attached is also helps us to understand how they move and why they move and what are they going for, or what are they trying to get away from. And so some words that you might hear as you go into your additional classes is words like monotrichous, lophotrichus, amphitrichus, or paratrichus. And basically mono, one, so that would be like a single flagella. Lopha, small bunches emerging from the same site. So that would be an example of the second one down here. Amphi, both, two-sided. So that's going to be like where they have flagellas on both sides. And then, of course, peri is around the perimeter. So they can have them all over. So again, this kind of helps describe their not personalities, but it helps us to understand, for example, why do women typically get more urinary tract infections than, say, men get? And what we're going to learn is that typically the bacterial etiologic agents, the things that are actually making you have the urinary tract infection, it really has to do with the bacteria, and those bacteria are really good swimmers. They have great flagellas and strong flagellas, and they're able to navigate and swim upstream, for lack of a better way of thinking of it. And so this is going to help us as we progress through the course, just to understand that understanding their physical structure helps us understand how they're going to affect us as humans. Remember a few minutes ago I said that they can either move towards or move away from stimuli. Well, we call that taxis. Okay, so taxis is to carry, and you can either have positive taxis or negative taxis, and positive would mean to swim towards, and negative would mean to swim away from. And sometimes they swim towards the light, or they swim away from the light. Sometimes they move towards a chemical. Remember a second ago I mentioned the urinary tract infection. Well, what we find is a lot of times your diabetic patients are going to have more UTIs than a person that's not diabetic. And why? That's because, think about it, diabetics are going to release glucose in their urine. Remember, glucose should be in the blood, not in the urine. And so these little bacteria are going to swim towards the glucose source. And so again, this is if you're wondering to yourself, why do I need to know this? It just kind of helps paint the picture of why these bacteria do what they do, and thus why do they make us ill? Why do we have problems with them? So let's look at this question. Let's see if we understand your external features, specifically your flagella. Which of the following best describes the action of a prokaryotic flagellum? Okay, so which of the following best describes the flagellas that are found on prokaryotes? Okay, number one, does it whip back and forth? Does it extend and contract to move the cell? Does it rotate to move the cell? Or does it attach to the environment and pull the cell? Or is it all of the above? Now, my hope is that you remember that it does not attach. Remember, the flagella is going to be for movement. So right off the bat, we know that we could pull out answer D, couldn't we? Okay, right? So if we can pull out D, that means E is also not an answer. Okay, so then that leads us to A, B, or C. And the key here is that with a prokaryote, does it whip or does it rotate? Okay, we know it doesn't stick out and stick in, right? We know it doesn't extend and contract, but does it whip or it rotates? And hopefully what you got from that process of elimination was C. It rotates to move the cell. Okay? All right, fimbriae. Again, external attachment or external feature. So what is it for? And I've already said this. It functions to adhere, to stick. So these are not for movement. These are for adhering to host cells. So when we think about, for example, E. coli, and again, I know that you don't think about this, but hopefully by the end of the semester you will. But E. coli typically gives us problems, right, in our intestines. Okay, so gastrointestinal issues. Well, one of the reasons that E. coli is so good at that is because E. coli is loaded with fimbriae. Okay, they're loaded with fimbriae. And this fimbriae is going to help, if you look right here in this picture, it's going to help the E. coli bacteria adhere, excuse me just a minute, it will help it adhere to our intestinal lining. And if you remember from your anatomy class, that microvilli, it's going to help it adhere to that microvilli. And remember that that's where the absorption of nutrients takes place, right, in our intestines. Well, guess what? They're for a free meal too. So they like to stick to those cells and thus absorb the nutrients that come traveling down our alimentary canal as well. Okay, pili. Pili was another external feature, and we know that pili are only found in certain prokaryotic cells. As a matter of fact, they're only found in gram-negative cells. So if you hear of something having a pili, we know that it is a gram-negative. Now, what is a pili? Well, just to be honest with you, a pili has often been described as, oh, well, is this like a bacterial penis? Is it like a bacterial appendage that is used for sex? And the answer is yes. It is not clearly, it is not a penis, but it is used for that same purpose. We know that it functions to join bacterial cells together for this purpose of what's called conjugation, and conjugation to conjoin, to come together. And basically one pili, right, from one gram-negative bacteria will insert into another cell, and it will transfer a little bit of its DNA, and we'll talk about that in a lot more detail later. But this is how that new cell becomes genetically unique, because now it has different DNA in it than it had to begin with. And so this is basically how bacteria or prokaryotic cells do sexual reproduction. Again, the only ones that have a pili are your gram-negative cells. Okay, last part about external features is going to be your glycocalyx. And the glycocalyx is a coating. It is usually made of sugars and proteins, so it's made up of carbohydrates and proteins. And if these carbohydrate protein molecules are tightly organized or tightly attached, it's called a capsule. And if they are loosely attached, it is called a slime layer. And so when we start looking at bacteria on our agar plates, sometimes the bacteria are in a very defined colony, and sometimes it looks very gooey, slimy, and so that would kind of indicate more of a slime layer versus a capsule layer. What is the purpose of this? It is to help cloak or hide or protect the bacteria in its environment, and so it kind of helps the bacteria to kind of go unnoticed, or maybe not noticed as much until it's had time to adequately reproduce and get some size on it. So again, I've already said that it's used for protection. It keeps the bacterial colonies or cells from drying out, dehydrating, and losing nutrients. It helps to inhibit phagocytosis, and if you remember phagocytosis, or if you've heard it called phagocytosis, either one, remember that's cell eating. So if you think about this for a minute, if a bacteria cell were to come into my body's immune system would see it and go, hey, you're not supposed to be here. You're not part of her cellular structures, and it would attack it. But instead, by making that glycocalyx, it kind of confuses my white blood cells and to thinking that, this is not a foreigner. And as a result, that cell is then able to multiply and get some size on it, and then my body would recognize it. But that glycocalyx kind of acts as a protection. And then lastly, it helps to form biofilms. And we're going to learn that helps to attach, and we're going to learn the importance of biofilms and why we have to control those, through our processes to make sure we don't get sick. Matter of fact, here's an example of a biofilm on a catheter. This is certainly why you would not reinsert, a catheter. Once you put it in and then take it out, do not reinsert the catheter, because a biofilm, those bacteria have been able to make a mesh-like structure on it, and then you would be reintroducing that. And you don't want to do that. That's going to lead to, to issues. All right. Okay, so I'm going to stop there for this video, and then I'll pick up with the next part.